Thank you very much for inviting me. I'm very happy to be here, and I'm happy to talk about some of this work, and thanks for a kind of nice introduction to what I plan to talk about from Charles Godfrey. When asked to give a talk here, I thought about, how are we using barcodes right now? And this is my title, and I will get to this in a minute. But in fact, my lab, the research in my lab has totally changed really with the DNA revolution, as it were, and partly because we could use DNA to reconstruct phylogenies, but using DNA means using barcodes at many different levels. And so one of the points that I'd like to make in sort of a conversation that I'm sure we'll have is that barcodes, the standard CO1 barcode, of course we've used a great deal of, but I think of many other kinds of barcodes now that we use for looking at different taxa. So it was about 10 years ago, I guess, that Corey Moreau came to my lab and we started to work on the phylogeny of the ants. The main point from this slide is really, or the main point from this tree was that the take-home message for us was first that the ants are very old, much older than people had thought previously, and Corey and I still stand by that. This tree was very well calibrated with fossils. But also, if you see this green bar in the middle here, that green bar represents the rise of the angiosperms. And you can see in this lineage through time plot here that the ants really diversified with the flowering plants. And that very much surprised both of us, I guess, because we had some prejudices about the idea of ants being scavengers or predators or so on. But flowering plants, of course, you can go back and you can say, with hindsight, yes, a more diverse world. They were feeding on insects that fed on those plants. There are many possibilities. We were also interested in bacterial associations with ants. And it was a suggestion from Nancy Moran in a casual dinner conversation that got us going, looking at the gut symbionts of ants. Nancy looked at me and said, "You have the genomic DNA, why don't you take a look?" And so we did. A postdoc came from Nancy's lab, Jake Russell, and he screened, and this was before next-gen sequencing, he screened hundreds and hundreds of samples. This was all done by cloning and sequencing. We had in that phylogeny two-thirds of the representatives or two-thirds of the genera of ants. So this was no mean feat. But he had a very nice result, which is that he found many bacteria that were in a specialized clade, specialized by ants. So there were Tetraponera associates here and Cephalotynia associates here. The convention here is this is the bacterial phylogeny, but it has the name of its host there. So these were clades of bacteria. They were in the order Rhizobiales, although rather very distantly related to Rhizobium, but in fact, here's where maybe my own limited microbial background at the time helped, because I saw this Rhizobiales, and I had been very interested in a report from Tetraponera of nitrogen-fixing bacteria in the guts of Tetraponera. And so we decided to use this Rhizobiales as an indicator to look at how this distributed across the whole clade of the ants. And as you can see here, Jake found 5 independent evolutions of this trait of having Rhizobiales on board, bacteria on board. But the other thing that characterized these groups of ants that had the Rhizobiales is that they were characterized using stable isotopes as being herbivorous or plant feeding. This means mostly feeding on plant exudates or nectar feeding. Nectar is rich in carbohydrate, very poor in protein. So we were amazed and thrilled when we found this very strong association right across the phylogeny of the ants. There are many hundreds of species involved in this. And what I would say is that we more or less stumbled across the legumes of the ants world. I have always thought of ants as being very much like plants. They grow like plants. They're sort of, as the workers go out and forage, they're sort of like the cells of plants. And indeed, we would say that having these symbiotic bacteria in their guts, so having the bacteria on board, may have facilitated this radiation of ants into nitrogen-poor environments, such as the canopy. So in these 5 groups, I think 3 of them are highly specialized to feed in the canopy in these nitrogen-poor environments. And it was another student, John Sanders, who has taken this work much further, studying the cephalotiny, the turtle ants, and their coevolution between these ants and their microbiota. And in this case, he was fortunate that recently a phylogeny of the cephalotes had come out, and his interest was to take the whole gut microbiota of the ants and look at how the whole community may have evolved across the phylogenetic tree. And he was very careful about how he did this. He characterized the gut bacteria for the cephalotes, both by using a fluorescence technique to see how many bacteria are there, but also by using qPCR to quantify how many bacteria are in the guts. Is it just a few, or do they have a lot of gut bacteria? And he confirmed very beautifully in results like too many I can show you that in fact they are chock-a-block with bacteria. And particularly these Veracue microbes, as you can sort of see plotted here, this is for 18 different species that were all collected in the Cerrado and in working with others in Brazil. I only make the point here that these are the 18 species, and these ones were ones that were taken out of ethanol. So you can see, and these were all had the gut dissected and looked at fresh. So you can see that there are some interesting ascertainment effects from how you pickle your ants, but that gets us onto another subject. So John characterized his bacteria in the guts of the cephalotes, and then he went on to look at how did they diversify across the phylogeny of the ants. So he had the phylogeny of cephalotes, and then he mapped the phylogeny of the bacteria. So here you see the bacterial phylogeny with the many different types, and the little blue bars here are actually sort of shorthand for the phylogeny of the ants. And wherever you see much blue mapped, it means that there was mapping of those bacteria onto that ant phylogeny. And wherever you see a red dot, it meant that he found a significant signal of co-speciation between the bacteria in the gut and the phylogeny of the ants. Now, this was an exciting result and still is very interesting to us because the age of cephalotes is about the same as the age of mammals. I mean, these groups are about the same age. And of course, for mammals, I'm sure you're all familiar with the work that's been done characterizing the microbiomes of different vertebrate and mammal groups. If this were a tree of mammals here, you would not see all these red dots. There's very little co-speciation of bacteria in the guts of mammals with their hosts. So something interesting and special is going on with the ants. We are also surprised because these gut bacteria are free living in the gut of the cephalotes. They are not endosymbionts that would be automatically vertically transmitted. They are free living in the gut. And we think that the way in which they are transmitted for cephalotes is actually a behavioral trait. The cephalotes engage in oral anal feeding like termites do. And we think that that's been essential in this vertical transmission of bacteria across, down through the lineages. I mean, this is this is an old clade, and so to see that signature of co-speciation is really something. And what John would posit, and I agree with him, and it's but it's still an open question, and it's really goes after the question that Paul Hebert asked. Is how do you? So we have bacteria in our guts. How can you recognize the good ones from the bad ones? Or how does the host recognize the good ones from the bad ones? Because some are pathogens and some are mutualists. And when we characterize the gut bacteria like this, how the heck do we tell the difference, even with our own bacteria? And what John would say is when you see this long pattern of co-speciation, it suggests that they've been associating for a very long time, and there's some stability to that over the ages. So those would be the bacteria to look at first for a possible mutualistic association. And I think it's a great hypothesis and worth testing, as he is doing, looking at the primate lineage, of course, because that's a case where we might actually have a chance of knowing what some of these bacteria are doing using metagenomic techniques. Okay, well, we don't always find patterns of speciation. Here's another example from my lab. About the same time when Corey came, Santiago Ramirez came from Colombia, and he was very interested in the evolution of euglossine bees and in pollination relationships and mutualistic relationships. He did a beautiful phylogeny of euglossines, and one of the great things about these little bees, of many great things about them, is that they give you some behavioral clues. So they carry pollinaria from the plants that they're pollinating on their bodies. And again, this was how barcoding of a sort absolutely revolutionized what we could do behaviorally, because Santiago could use this barcode, and in this case, we needed 2 genes to be able to get or 2 gene markers to really go for this. But he was able to then capture many bees that had their pollinaria on their bodies, and here you can see all these little bunny ears. They're very cute. This was the group of pollinaria, and come up with a matching to the orchids that they belonged to, and then look at how that matched with the phylogeny of the bees. Was it one, really, if you go way back in the textbooks, it was thought to be an elegant one-to-one matching with an orchid pollination, pollinaria to orchid. I don't think we would think this now, but we were very surprised at how reticulated this pattern was. There really didn't seem to be much one-to-one matching at all here. But there are some very interesting things that came out of this. One thing that you might notice right away is that the bees are much older. The bee clade is much older than this orchid clade that we're looking at. That means that these bees evolved and were in place long before the orchids began to diversify on that template. And I would say that this is has been a common theme that we've seen in many mutualistic interactions of this sort when we've compared phylogenies. That is, one partner tends to be older and more robust than the other, in the sense that these bees may be able to live very well without the orchids, but the orchids can't live very well without the bees. Indeed, you can think of the bees as being a sort of ecological template against which the orchids have diversified. And this is also true, for example, if we look at the ants and the lycaenid butterflies whose caterpillars, many of the caterpillars of which associate with ants, they too have this kind of asymmetrical pattern of dependency between the two groups. Moving on to another example that goes more into community ecology, a current student, Leo Biddleston, has been looking at the communities of pitcher plants. I particularly like this study because it has a sort of a ground truthing to some of the molecular work we do. So she took 3 species of pitcher plants here found in Singapore, that wonderful wild field site. But I think she was waiting to get permits to go to Borneo. But so she sampled these 3 species and looked at what was in their in the pitchers. This is a much deeper study, but I'll just talk about the insects because this was so much fun. So you can look inside the contents of the pitchers and see the carcasses of insects that have fallen in there. Some of them are inquilines that actually live in the pitchers and eat the prey, eat some of the prey that the plant has captured. And when we look down, we can count those morphotypes for those insects in there. And there are, in this particular group of pitcher plants in Singapore, there are 8 morphospecies. Well, she was able to then take the fluid from those pitchers and use meta-barcoding to look at how many arthropod OTUs, or operational taxonomic units, were inside those pitcher plants. And she found 189. So it was a pretty good, to me, I thought, oh, that's possible. We're seeing the traces of 189 arthropods that have been in or around, maybe digested by the plant or related in some way to the inquilines inside those pitcher plants. But the nice thing is that for some of the taxa where we could identify the morphospecies, there was actually a reasonable correlation between the number of OTUs for that taxa and the counts that we made just by taking the pictures ourselves and counting the morphotypes for the insects. Although you can see here that mosquitoes and mites tend to be underrepresented in the data, but many of the midges have a very good correlation. So it's a kind of a ground truthing for some of our molecular techniques. She also did some ground truthing for the identification using barcodes from the 18S data for eukaryotes. She also sequenced CO1, the standard barcode, to make sure that, and indeed there was a very, I think, quite a good matching between the insects that were in there and the way in which we identified them. Of course, she's looked at many other things in here. I can't go in discuss them all, but just to show one of these nice bipartite plots, as Charles Godfrey did already, this shows rather nicely, it's plotting the arthropods against the pitcher plants, and it shows that there's a significant association between the species of pitcher plant and the community of arthropods associated with it. And you can see this perhaps a bit better in this spring-loaded diagram that gives you a sense of those OTUs that are unique to a given species and the ones that are shared in the middle. Okay, well, I said I would talk about Myrmecophiles, and I will now in the last 10 minutes or so. We're now moving to Africa, and to the entomologist's delight, because you can see these balls hanging in the branches of the acacia tree. These are the swollen thorn demacia that the plant produces for the ant. This is the work of current students, Dino Martins, who recently left, who really brought the whole system to my lab. Dino comes from Kenya and emailed me and said, well, you'd better study this, and I had to agree with him. Chris Baker, who has done a huge amount of this work on Myrmecophiles and on fungi, I'll talk about in a minute. Jack Boyle, who's been working on the population genetics of the ants. Julianne Peliez and Laurie Shapiro, who've been looking at some of the chemical ecology involved in helping with the field work. It's a great pleasure to talk about ant plants and mutualism with Dan Jansen in the audience, because a lot of us were inspired to work on mutualism because of his pioneering work years ago, looking at using ant plants as a model system for studying mutualistic interactions. There are many, this is a plant trait that's evolved many times in many different species of plants. And yet they are all restricted to tropical areas, an interesting factoid that I'll come back to at the end. Here, going to East Africa, it is tropical, but it's more a savanna. We're entering into the savanna where we evolved as a species. And here is Acacia drepanolobium, or now called Vachellia drepanolobium, with its swollen thorns and antimacia. This is a dominant plant throughout East Africa as far as the eye can see. This is looking across the Rift Valley. Wherever there are black cotton soils, you see these Acacia drepanolobium, or Vachellia drepanolobium. Up to 18 ant species have been recorded living in those swollen thorns, but really there are, across the most populations, there are 3 dominant phytoecious ants. Phytoecious means that these species are only found inside this ant plant. They're not found free living. There is one other that's located in Impala that is a free living one. And it's a straightforward house and food in exchange for defense against herbivores, mutualism. There's been legions of work done ecological work done on this system by Truman Young, who really started it, Mo Stanton, Todd Palmer, Rob Pringle, and others who have looked at some of the costs and benefits of associating with different ants in the system. So that was very helpful when we started. It's still helpful in being able to study aspects of it. Dino Martins did a beautiful work where he showed that the ants, even these very small ants, can be extremely effective against these rather large browsers like the giraffe, which approach the plants inhabited by the 3 different ant species equally, but will not browse on the one with Chromatogaster mimosi, which is the most pugnacious of the defenders. This wimpy Tetrapona apenzigi never does a thing and seems to not deter the browsers at all, and has been quite a mystery. Okay, so this system has many third-party associates. We, of course, became interested in this, not the least because if you open up some of these Demacia, you can see here these 2 nice polymorphic lycaenid caterpillars inside. The caterpillars of many butterflies in the Lycaenidae associate with ants. They can chemically fool the ants into favorable recognition, where the ants will actually tend and protect them instead of recognizing them as potential enemies. And there are many lycaenid butterflies that parasitize this ant plant system. Here are just 4 of them, but Dino and others have documented up to 45 different species. There are weird and wonderful flies in there. If you open up these, the Demacia, there are also xeraptorins that feed on fungi inside some of these Demacia. There are beetles and flies that range from being mutualistic, apparently mutualistic in their interactions with the ants, to parasitic. So we wanted to do a survey to see, how much is really in there, because all the focus so far has been on these large browsers. What about the things coming from inside? And to do that survey, we went out and cut down small plants, took all the Demacia off them, and sorted them, and then wonderfully, because in the past, these are mostly larval forms that we were finding in there. In the past, we would have never been able to identify what these things are. But of course, now we can. So by pulling out OTUs or barcode sequences and then blasting them against different databases, we're able to identify what is inside these Demacia. So here is our data set from 2 field sites in Kenya, Kitengela, which is outside of Nairobi, and Suyan, which is in Laikipia, up near Impala, for those of you who've been there. The results of searching through about 22,000 Demacia, we ended up with about 2,000 some Myrmecophiles, and from those identified 82 OTUs. How did we do? Well, we estimate from this rarefaction curve that we probably have identified about half the OTUs that are present. So maybe there are 160 to 200 species there in this system, or that's what we would estimate that's from 2 main field seasons of work. Here are some of the critters. We've taken them out and in all cases photographed them as well, and of course looked at various aspects, much of which I can't talk about. Are there more Myrmecophiles with one ant species than another? And yes, Mimosi, even though if you control for a Dalmatian number and so on, it seems to have a much larger burden of Myrmecophiles than the other 2 ant species. So I hope you see right away that there's an irony here. This is the most pugnacious ant that keeps things from browsing, and yet it's the one that's sort of parasitized the most by Myrmecophiles inside the nest. Myrmecophiles many of which are carnivorous inside the nest. Now we can characterize the ant communities in different ways. This is just a canonical correspondence analysis that gives an ordination separating out the Myrmecophiles. Each one of these dots represents a tree, and this pulls out the ones for the different, inhabited by the different ant species so that we can better look at their ecology. And indeed, there are significant communities of Myrmecophiles that are specialized with particular ants. And just to give you an example of a few of these, there are some generalists. So this Gellichiid is a generalist, and so oddly is this ant mimic. But then we have others that are specialists. Of course, the scale insects are huge specialists with Mimosi. And interestingly, the gastropod with Pensigi and Tortricid there is specialized on the chromatogasters. So it's an ecological playground here to look at some of these differences. We can, of course, also use the barcodes to establish life histories. So David Agassi had written a very nice paper where he described different moths that he'd reared from the acacias, but without taking into account the ants that associated with them. And we can now match the larval forms of those moths and their larval and pupal forms. Okay, well, much too much to talk about, and I will move quickly on for the Myrmecophiles, though, simply to say that there's a great deal of variation, but we do get interesting specialization and on different ant hosts. But that's not the only thing that's inside these Dalmatia. And one of the reasons we got interested in looking at one other feature in the Dalmatia is because of the wimpy tetraponar ants that I mentioned before. Dino had long wondered about them because he never saw them out there protecting. He never saw them attacking a herbivore at all. And yet when he reared them in the lab, they showed an uncommon interest in fungi and in harvesting fungi. I'm almost done. And indeed, here you can see that if you offer them fungi, they will harvest a great deal more than the other ants. Well, of course, we could use the same amplicon sequencing techniques to look at what was in there. I like to think of the different ants on the different plants as exhibiting a sort of extended phenotype, where the Dalmatia itself becomes like an extended phenotype that's good for growing particular cocktails of fungi. And we could sample these and also the ones that we grew in our greenhouse that was outside of the spore reign of fungi available. And here you can see that we got very big differences in diversity. So the leaves have the greatest diversity of fungi, and the greenhouse had almost none. This is a greenhouse in North America. But what about the communities of fungi? Do they sort out by ant? And the answer to this is yes, absolutely they do. We got Tetraponera has a very typical community that's separate from the Nigerceps, that's separate from the Mimosi. And we can even twirl them around and look at them this way. We can culture them too. Here's a bipartite graph, again, showing you that there's specialization and that we can culture them. These are the ones we have in culture, and what we would like to do now, of course, is go back and see what effect do these fungal communities have on the plants that they're in. I add one last thing, which is that with Johann Billen, we looked at the infrabuccal pockets of the ants and found that the alates of, in this case, nigroceps, were carrying all of the fungal diversity that are found inside the domatia. I hope you know why we find that exciting. How does the fungus get into the domatia in the first place? Well, the queens burrow their way in. Okay, so the take-home message from all of this is that we really need to think about this mutualism. Now, if we think of it as a guild of interacting species, we need to take into account these third parties. I gave the example of mimosae before, but here's something to go home with and think about. Let's imagine that what the real mutualism in ant plant mutualisms is actually between the fungal community and the plants. If the fungal community inside an ant plant encourages it to grow better, then the ants may simply be a vector to carry that fungal community. This is a mutualism enhancing mutualism. And of course, ant plants are only found in the tropics. Maybe that has, and as Doug tells us, the more tropical you are, the better you are at having fungi present in wood. So perhaps there's something to that. Okay, and I will end now with a note on barcodes, which is that they're fantastic identifiers because that you can use them to identify both pattern and process. And the reason you can is because since it's DNA, you can use them both as markers to identify things, but also to reconstruct phylogeny and evolutionary history. And I know I'm preaching to the converted here on this, but we've been able to use that in many different ways in my lab to characterize these symbiotic interactions that undoubtedly have promoted terrestrial diversity. And with that, I will stop. So thank you very much.